Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to this thought leadership interview on the topic of biosimilars for OCT USA 2020. I'm your presenter, Rick Navarro. Today, I have the pleasure of interviewing Associate Professor Paul Griffin on the subject of biosimilars. Paul is a Principal Investigator at Nucleus Network and one of Australia's most renowned infectious diseases experts. He has personally overseen more than 100 clinical trials and is currently the Principal Investigator of 3 separate Phase 1 COVID-19 vaccine trials at Nucleus Network. Paul, welcome and thank you for joining me today.
Thank you for having me.
Now, Paul, I'm going to launch straight into our topic of biosimilars. We often hear the terms biologics and biosimilars intertwined, so right off the top, can you outline the difference between the two and give me the elevator pitch on what exactly are biosimilars?
Yeah, it is a bit of a confusing terminology that we use for this. So a biologic is a medicine that's basically made from or contains something made from some kind of living thing. So one of the the oldest examples is insulin, which I think is around 50 years old now, which is something that was made from cells and we still use to this day. And they're really fast growing. So we've seen now, I think, around 300 biologic drugs that are in use at the moment. And they're quite complex. So they're often large proteins, as I say, made from living cells through quite complicated manufacturing processes. So when they come off patent or we're trying to improve on them in some way, it's not as easy as with small molecule therapies as just making another one. So that's why we have to have what we call these biosimilars, which we think are very similar, but just can't make exactly the same way. And that's why we need to have trials to show that they are indeed the same and have a very similar, if not the same sort of action.
So why is it that you think that we're starting to see an increasingly large volume and number of Phase 1 biosimilar clinical trials?
It's a really rapidly growing area. So I think it's the fastest growing area of therapeutic compounds. And obviously the type of conditions that they treat are things like autoimmune or inflammatory conditions. And we're seeing the rate or the number of those conditions go up as well. But we're also seeing increased capabilities in terms of the biologic drugs that we can make with all sorts of properties that improve on their predecessors. So, and we're also seeing a large number of biologics that are coming off patent, and that's why the biosimilars that are the same or perhaps improve in some ways on the properties of those biologics that are coming around.
So continuing on that theme, are Phase 1 biosimilar clinical trials different to your typical small molecule first-in-human healthy volunteer studies? And if so, how do they differ?
I guess the main thing to say is that the key processes are still the same. So we still want to do high quality clinical trials that are based on robust preclinical data so that we make sure we're not exposing our volunteers to any undue risk. And we want to make sure that safety and quality are the focus of those trials. But there are a few differences. I mean, obviously we need to have comparable PK and PD, so pharmacokinetics and pharmacodynamics to the reference product. And there are some slightly unique challenges in terms of things like immunogenicity and anti-drug antibodies that we don't see with small molecules or traditional types of therapy. And the other thing, I guess, is that a lot of these biologic products do have interesting mechanisms of action and outcomes. So sometimes there are challenges doing those in healthy volunteers. But the other thing is they're often quite long-lived in terms of their effects. So we need to do fairly long trials, and that presents some fairly unique challenges in terms of being able to recruit to those, but then also retention. And I guess that's where units like ours that do a lot of this get some special expertise in those areas.
So let's go a little bit deep dive in terms of biosimilar clinical trials. What are some of the specific challenges you're observing, and how do you address them when undertaking biosimilar trials?
So yes, certainly the length of the trial. So we need them to be fairly long trials, and they're also fairly large in number, often around 200 or so volunteers. So therefore recruitment can be a bit of a challenge, and we need to make sure we've got good processes around that, and that's something that we've worked on over the years to make sure that we can do that and do that successfully. And a lot of that is just about having some skills in the area and making sure that we recruit the right type of volunteers, and we make sure we educate them well and look after them well, so that they not only enroll in the trial, but then also stay on board as well. And because a lot of it is done via outpatient visits, you need capacity in the outpatient space to be able to see those people and see them effectively and efficiently, so that, again, they keep coming back and retention is not a problem. So having a large capacity for outpatient visits, which a lot of phase units don't necessarily focus on, is also a big key component.
So you've touched on quite a few things there. Are you able to bring any of those to life through a recent example that comes to mind?
Yeah, I guess I don't have one example. We've done so many of these sort of trials in recent times. So certainly around Herceptin, we've had some quite a few in that space, as well as Neulasta and Xolair. So we've done quite a few of these trials in recent times. So it's hard to pull out any one to be the example here. But with all of those, I think it's really been about having the experience and the expertise to make sure that we can fully recruit them and then to retain those subjects. But also, I guess it's the key concepts that underpin any clinical trial, which is really about the safety and the quality and having, really high levels of attention to detail to generate robust data whilst making sure we look after our volunteers really well.
Excellent. And you touched on it before. I'm going to get you to expand on it a little bit more now, and that's the requirement to have specialist infrastructure and support services to effectively conduct biosimilar phase 1 trials. Can you expand on that a little bit in terms of what you mean by having adequate infrastructure?
Yeah, so I think in terms of the actual physical infrastructure, it's really the outpatient capacity that is the key with biosimilars. So because a lot of the visits are done on an outpatient basis and because they're typically fairly large clinical trials, again, that sort of 200 odd mark, you need to have a lot of beds. And again, if we want to get people coming back for all those visits, we have to do that really well, because if there's a lot of waiting or congestion, then that's going to really affect our retention. So, having a lot of focus on retention and making sure we do those outpatient visits, really effectively and efficiently so that they keep coming back is really important. So, yeah, it's really about having outpatient capacity, I think.
Yeah, good key point there. So understanding all the challenges you've outlined and considerations for a biosimilar phase 1 trial, how does Nucleus Network successfully recruit for participants, particularly given the long duration and stay that's required over a long period of time for our biosimilar trials?
I think it's about focusing on end-to-end, so all aspects of the biosimilar trial. So, obviously in terms of, getting them up and running, having a team that's really experienced in the type of regulatory hurdles that you might face with these sort of products and making sure those regulatory approvals can be done quite quickly as well as effectively. Obviously, the recruitment team come into their own and, an experienced recruitment team that, again, are used to having the sort of conversations you have around these sort of studies with a very large database from which we can get some volunteers and certainly also some dedicated advertising potentially, depending on the therapeutic area or the condition the biosimilar might be targeting. I guess it's then the clinic staff, both the inpatient and the outpatient staff, the medical officers and the principal investigator, having general clinical trials experience and expertise, as well as some expertise in the biosimilars, so they can face some of these specific challenges. And then the guys in the outpatient clinic with a real focus on retention. And we have some specific plans around subject retention that have been developed to specifically address some of these challenges and to do that really effectively. And, we take great pride, particularly in the safety of our volunteers, but also the quality of the data we generate. And if we don't get a volunteer from start to finish, attend all the visits on time as scheduled and get all the information, that the PK data, then obviously that's going to potentially undermine the study. So, we make sure we get the information we need when we need it, so we can complete the study as we intended it to be.
So notwithstanding that sort of specialisation and skill set in participant recruitment, but also retention, what are some of the attributes that make for a successful phase 1 biosimilar clinical trial?
So I think it's all of those things, really. I mean, there's attention to detail is a really big attribute. Again, to make sure that we get the data we need to be able to show that the biosimilar has the PK and PD that are comparative to the product from which it's based is really important. And so, it's really about the attention to detail. And I think outside of phase 1 units that do this all day, every day, the understanding around that level of detail just isn't there. So, again, that's something that we take great pride in doing, is making sure we collect those samples when we intend to, and that they're processed exactly as they are intended, so that we can get that data to support the approval of that biosimilar product. So, as I say, it really comes down to, I think, experience in clinical trials generally, and the same sort of things that underpin clinical trials, but with a particular attention to the key elements, which is about getting that data we need, which is subject retention, but also quality and attention to detail.
So you've covered quite a bit of really interesting terrain here. So I'm going to get you to potentially summarise all of this in what would be your top top line 3 considerations that you think are necessary for a successful phase 1 biosimilar clinical trial. So how would you summarise that in 3 key takeaway points?
I think it's about understanding the intricacies of biosimilar trials, and particularly the longevity of the trial and the fact that it's so outpatient focused. So I think having the infrastructure to support those outpatient visits, and also having the expertise. And again, it's a multidisciplinary approach from the recruitment team to the nurses to the medical staff, who again have experience and are focused on the biosimilar and making sure those subjects keep coming back so we can get the data we need to do that study well.
Yeah, that's well summarised. Thank you. So from what I can glean, Paul, we're on the cusp of a new wave of biosimilar trials, and your insights have certainly provided a fascinating roadmap to really understanding the intricacies and the successful, I suppose, realisation of biosimilar trials. So thank you so much for your time and expertise.
It's a pleasure. Thank you very much.
If viewers have further questions, please stay with us as we answer those for you now in our Q&A session. And also, be sure to come back and check us out at OCT USA, our virtual booth, where you'll find a plethora of information about Nucleus Network and our unique offering as the only phase 1 clinical trials specialist with clinics in both Australia and the US. Thanks for joining me in our quest for advancing medicine and improving lives. Cheers.